Okay, Act 2. All right, let's go. All right, so, okay, to answer, can I just answer Priyasha again? Let's go through it again.
Okay.
But now,
Just lift up the...
no, that's fine. Okay. Let's just go through it one more time. We don't need that. And where's my little goodie? Okay. Go away. Go away. Okay. All right, so let's just, you're going to tell me now, all right? Okay, so, and I'll come to your question about counting stuff. Okay, so when we're talking about human cells, but when you get a question, it might not be a human cell, because to draw 46 chromosomes is going to be a mission. Okay? But a human cell, a normal body cell, something I didn't mention just now, a body cell, oh, this is a horrible thing. Okay, a body cell is called a somatic cell. Just don't write it down. Somatic. Then you've got the gametes, which are haploid. Okay, so body cells are diploid. In humans, they've got 46 chromosomes. When the cell isn't dividing, we call it interphase. But then at the end of interphase, the cell starts preparing for cell division, and one of those preparatory stages is the DNA becoming more organized and replicating. Okay? All right. So at the bottom, we were just saying dogs have 78 chromosomes in their cells. All right. So when the cell is in interphase, the DNA is more thread-like. We call it chromatin. But now it's going to start preparing for cell division, so it starts coiling up. It's still DNA, but because it starts coiling up, we call it a chromosome. Then replication happens and nucleotides get added. So you end up with how many DNA molecules? Two. Those two DNA molecules are called chromatids. And they're held together at a region called the centromere. So now, when this replicated chromosome splits, okay, so this is a replicated chromosome. It's still one chromosome, but there are two DNA molecules or chromatids held by a centromere. So in mitosis, do you remember karyokinesis?
Yes.
That's when the DNA gets separated into daughter cells so that you end up with two nuclei. And then they will both have how many chromosomes in the case of human cells?
46.
Good. So here, when all the chromosomes have replicated, there's actually 46 times 2, which is how many? 92 DNA molecules or chromatids. But how many chromosomes are there?
46.
But they replicated.
Boom.
Okay. Yay.
That makes so much more sense.
Kayla?
You answered my question.
Okay, Priyasha, did I answer yours?
Yes, ma'am.
Okay, that's the confusing part. So we do it again and again and again in grade 12 as well. Okay, we got it. So let's now look at the stages of mitosis. Yes?
Oh, no, I was going... Sorry. I don't know what you said.
All right, so let me pull up. I'll have to move that now. Wait, I've got some more. I don't want to really spend the same amount of time I did doing mitosis online. Because to me it's done, but
The phases were pretty easy to
Yes.
understand.
it's this part.
Yeah, exactly. I understood the phases perfectly.
So we'll just revise.
One more time.
Yeah.
Yeah, we'll just revise the thing. So, okay.
Telophase.
So here, okay. So over here, so there's that replicated chromosome with two chromatids. I mean, they draw it funny with this big blob. It's actually not even a blob, it's just a region where they join. And there's a proper chromosome. It's very tightly coiled DNA. Look at that. Twist, twist, twist.
So then this one is a replication, this one is a replication.
So, this is a elastic band very twisted. This is a DNA molecule very twisted, held together by centromere.
I thought it was 4 for some reason.
1, 2, held together. Oh, you know why? That's why I always do that. 1, 2,
Yeah.
but it's actually 2.
2, but it looks a little like 4.
But it looks like under an electron microscope. Okay? And then what they do is they take cells. So say you're worried that your baby's going to have Down syndrome.
How do you get
I
Down
did it with
syndrome?
Madam because I was older. I always tell the story, you're just going to get used to it. Okay? So, yeah, grade 12 we do amniocentesis and all of that. So, because I was older, over 35, yes, I'm old. Yeah. So, they take, because you know the baby's surrounded by fluid,
Yes.
and that fluid has got skin cells from her, just like we lose skin cells, and it's the dust around our house and gives us allergies. Okay, so they take some of the fluid, it's like a yellow color. So they numb it, and remember they're numbing the area, and then they push this like a big needle in, and then they draw out the fluid, and it's yellow, and then they send it off to a lab, and shame they can't see me. Send it off to a lab, and then they culture the cells. So there's cells in that fluid, and they give them chemicals and nutrients that allows them to start dividing. And then when they get to metaphase, those are the chromosomes that have lined up along the equator, and they take a photograph. Now, I remember many years ago, when I was at my previous school, we used to take grade 12s to the Mayo Clinic, the genetics lab. We didn't have copy and paste, so they used to have this photograph, and they used to cut out these chromosomes and then match them up. All right? And that's a picture called a karyotype. So we do that in grade 12, but it's nice to see now. So there, look. Chromosome pair 1, pair 2, pair 3, all the way up until pair 23. But here, they actually are double. They're just so close. The chromatids are like that. They are actually double. Each one there is a double. So both of those are double, but chromatids are just close. And they match them up according to banding patterns. Some of them are all curled, they're all coiled over and stuff. That's an electron microscope. This is I think it's a very high-powered light microscope. So they take the photo, now it's just copy and paste. And then they pair them together, so you get a picture that looks like this, and they put them all together based on these little banding patterns. They stain them. And I don't think I'd be able to do it, but each little banding, each little dark and light, it's not a gene, it's a long section of the DNA that just takes up the stain. And then they match them up. So in the case of Down syndrome, when they do this, they would have found that there would be an extra chromosome 21. And because then they phone and say, no, everything's fine.
So how many chromosomes does a Down syndrome person have?
Okay, so this is a normal karyotype. Is it male or female? This is just interest now, people. Is this male or female?
Male.
There's male. So there's a long X and a little Y. Okay? In the case of females, we'd have 2 X's. But in the case of a child that's got Down syndrome, there would be another one here, 21, 3 pairs. So it's called trisomy 21.
So it's actually, I thought, what's the one that if your child is held without oxygen for a while, that they get?
Yeah, but
Like if the umbilical cord is around their neck for a long time and then they're born, they have I thought they had Down syndrome, but clearly not.
it can cause mental deficiencies because the brain wasn't getting oxygen. But genetically, they would have still been normal. But here, because of that extra whole chromosome, it causes the symptoms and you get Down syndrome on a whole spectrum from very mild. Most people, a lot of Down syndrome people have medium symptoms, and a lot of them hardly have any. So there's this whole spectrum, just like from short to medium height to tall.
Oh, that's so.
There's a whole range. And some Down syndrome embryos, remember the little embryos, that people don't even know they're pregnant and they've already miscarried. Because there's too many chromosomes. You see, in humans and animals, if there's too many chromosomes, it's a problem. Sometimes if there's a gene that's wrong, it's a problem, and there's a miscarriage.
So, ma'am, if you have a miscarriage early on, so when it's just an embryo, will there still be blood?
There might be, and people think, oh, I started my period again.
Yeah.
They don't know.
That's so sad.
Okay, anything else? All right, so that's what I wanted to show you. That's what the chromosomes, that's what they look like at metaphase. They're lining up along the equator. And that's across the equator. You're looking at the equator from the top, like here. They're lining up here, and then half the chromatids get pulled to that side, and half of them get pulled to this side. It's an imaginary region in the middle of the cell. Okay? All right, so.
I was just kidding.
Now, this one, we don't do S1, G1, it's just too much. So the little diagram in your book where most of the cell's life is spent in interphase, the blue. Alright, so the cell grows, and then it's going to start, if it's going to divide, it copies its DNA, and it copies, if it's an animal cell, it copies its centrioles, and it makes more mitochondria. It's getting ready. Okay, it's going to split.
So they do copy their mitochondria and all that.
And other organelles, yeah.
So
So that all, so here's normal, where they're making more proteins, and then they start copying their DNA and their centrioles and etc. And then the actual stage, there's too many here. The actual stages of the nucleus dividing is called karyokinesis, and it's, okay, so interphase, then comes prophase,
metaphase, anaphase, telophase.
By telophase, there are two nuclei, but the cell hasn't divided, and when the cytoplasm splits, it's called
Cytokinesis.
Okay, so it's actually IPMAT cytokinesis. Interphase, normal cell function and preparing DNA. Prophase, metaphase, anaphase, telophase. Nucleus, the DNA being separated into daughter nuclei. Cytokinesis, cytoplasm.
Do we need to know karyokinesis?
Yes. Okay. All right.
Karyokinesis.
Karyo, kernel, kinesis, split.
So it's just all of them except cytokinesis.
So there's that little embryo. The little buds in the heart and the brain and the eye starts developing. And then you've got, I think I put this in because I said, well, you've got mitosis. And the one year I had a comprehension on apoptosis.
What's that?
So we've got mitosis to help us grow from a bundle of cells to an embryo. But then, remember, how do your finger, you've got a paddle. Look. So now hang on, we've made this hand by mitosis. So how do we get separate fingers? Programmed cell death. Apoptosis, the cells in between die.
Oh. So are there people whose hands are
So
like
if
this?
people like have webbed hands or webbed toes, did it not like?
So-
Can't you just?
So the gene for causing cell death, the gene for causing programmed cell death didn't switch on, and then the fingers stay joined.
Can you be born with a tail?
Yes,
Can they cut it off? Though?
yes.
Oh, how?
Because it doesn't get absorbed. It doesn't get absorbed.
Isn't that some of your spine being cut off?
They've got extra, and like a dog has got extra vertebrae,
Oh.
so they take it off. The spinal cord usually doesn't
And
go
is it
in
like a
there.
proper surgery, like?
It's sometimes it's just cartilage or whatever and tissue. Other times it might be, I think, might be little vertebrae in there, but hasn't got the spinal cord. It's just like it
Oh.
stayed there.
Okay.
Okay. Well, I haven't read up a lot on it. Okay. All right.
But look at the head in the beginning. Wait, now, can you just go back? Look how big the
And
baby's
there's the
head is.
face. Yeah, because the brain develops
What's
first,
that thing in
and
the
the
middle?
heart. Umbilical cord. That's the umbilical cord. There's the little buds. They become the little hands, and the umbilical cord is in here. Okay.
Now, what's the himblida bud?
Hind limb bud. That becomes. these
Is
little
that
buds.
the tail?
That's the tail. So that part that disappears.
It's so big.
So that's apoptosis. That breaks down. These become the hands and the feet. His eye's going to be here. Brain in there. Heart you'll see very early on.
I don't want to have a scan until the baby's proper.
This will be a block.
Oh, okay.
This will be a block. All right. So yes, you do need to know karyokinesis because. Haha. Okay, this is for the kid, grade 9, to point at the board. Okay, nothing else. Yes, interphase. So the cell prepares for cell division. The DNA molecules replicate. Then you've got karyokinesis where the nucleus divides. That's where these chromatids are now going to separate into daughter nuclei. So prophase, they're still coiling up, etc., and the nuclear membrane disappears and the nucleolus disappears. Metaphase, they're lining up, remember, along the middle, okay, along the equator. They're lining up, and then the spindle fibers attached to them, and then anaphase, A for apart. Okay, so prophase prepared, metaphase middle, anaphase apart, telophase 2 nuclei. Then the cytoplasm divides. Okay, all right. And there we go. So I gave you this one, but I've got others, but anyway. All right, so yeah, what's this? This is interphase, and then somewhere between here the DNA replicates. There you can see the chromosomes are replicating and they're starting to coil up. There the centrioles, only animal cells, have replicated, move apart, spindle fibers develop between them.
So so it replicates before it condenses.
Yeah.
I don't know.
But don't worry about stuff like that, because in other books they might say it happens at the same time.
Oh, okay.
The cell isn't saying, okay, the humans say I must now do this, then I must
Okay.
stop doing that, and then I must do this. It kind of happens.
It just happened right so.
Yeah. Okay, so there the chromosomes are called up. What is the diploid number of this cell?
4,
4. How many DNA molecules are there?
8,
Good. How many chromatids are there?
8.
You count, yes. You count chromosome numbers by counting this.
Centromere.
So how many replicated chromosomes are there?
4.
Okay, hang on. Yeah, so there's 8 DNA molecules. Each one is a DNA molecule. They're called chromatids, but there's still 4 chromosomes, but they just replicated. Not 8 chromosomes. There's 4, but they've got these 8 DNA molecules or chromatids.
Matt, I was meaning to ask, in some of the pictures like this that you've given us, there are a couple of steps that are different.
Yes.
That they say this thing happens in prophase, and then another one will say that
Yeah.
only happens in metaphase.
Don't worry about it.
In a test, if we mixed up those simple steps, and
Yeah.
would we lose marks for that?
So it always says use the picture to say.
Oh,
Okay, always does that. All right, and okay, so now nuclear membrane breaking down, the nucleolus, there it was breaking down already. I couldn't even see it yet, maybe it's behind. So now the spindle fibers have formed, and they're going to attach to the chromosomes, and as they do that, then these chromosomes start moving to the equator. So this is metaphase middle, centromere split, like pulling the Velcro apart, and then those spindle fibers, little protein fibers, start contracting, and they pull the chromatids, or now we can call them daughter chromosomes. And they actually do move in a little V like that, because they've been pulled. It's like pulling someone by their hair and their legs. Okay? So they do move in a V like that. So half the chromatids should move to one pole, half to the other. The nuclear membrane reforms around each group at the pole and the nucleolus. So that now is end of telophase. Then the cytoplasm dividing, easy in animal cells, is called cytokinesis, but in plant cell that cell plate has to grow across. The cellulose is deposited, and then eventually the cell splits into 2. Yes?
Since plant cells don't have,
Centrioles.
yes, how does the spindle fibers work?
Okay, so they don't really know. Okay, now we always draw animal cells round and plant. Yeah, they can see. Plant cells. Can you see though? Okay, so okay, let's get rid of this. All right, so we always tend to draw, even my posters there, plant cells, rectangular, animal cells, round. But you've seen, differentiation.
Yeah.
Animal cells can be spindle-shaped muscle cells, nerve cells with sticky outputs, etc. All right, what was the question? Oh, so in plant cells, it's like a little anchor point that develops. You don't need to know, it's called the aster. And then the spindle fibers develop from there, and they attach to the chromosomes. It's like a little anchorage point. And they don't really know. I've got a book in the back, which they made us buy when I was studying. A long time ago. It's this thick, and it's only on the cell. I'll show it to you.
Damn.
Okay, so they don't know why. They're still learning about the cell.
So I was going
Oh, my nose.
yeah.
Okay, so let's see what I've got on after this. I must attach my face, but my nose is itchy. All right, so now, did you do these? Okay, so what stage? Where's my stick? I think it's better. Okay, right. What stage is this?
interface.
Yeah, possibly. Okay, yeah. Oh, it's written here. It doesn't matter.
Prophase.
Can you see how the chromosomes are becoming more distinct? All right, and then that would be anaphase. This is? They don't look like they're nicely along the middle. They're somewhere in the middle, and I think that this is one cell, and you can see that cell plate forming.
Oh, telophase.
So that's telophase and cytokinesis of the plant cell. All right. Okay. There's plant cell interphase. There's no centriole, see? But you can see the nucleolus. There's the DNA. Now it looks granular because when you cut through a cell, the chromosomes aren't going to line up there nice and say, okay, take a picture of me. They're going to be long DNA molecules. You're looking at the ends of them. It's like looking at the ends of, well, they're not going to line up neatly for you to draw a pretty picture. You're probably looking at where the DNA molecules have been cut. So that's why it looks very granular, okay? All right, so then plant cell prophase. Now you can see they're becoming more organized.
Let's get together.
There's metaphase. There they're lining up. There you can sort of see spindle fibers. All right, there anaphase later, being pulled to the poles. Now you can see the cell plate.
Yeah.
So this is telophase and the beginning of cytokinesis in a plant cell.
So they kind of all merge together. All
Yeah, so they don't say, okay, when must I start and stop the next phase? It happens as a flow. All right, sorry guys, I need to go off. So here now, okay, there's lots. So what's this one? Okay, so it's plant cell, interphase. Look, granular. What is this one? But it's very dark.
Oh, yeah.
Probably prophase. Okay, you can't see everything that we draw. What is this one?
Anaphase.
Anaphase, there.
Metaphase.
Metaphase. Now, how do we tell telophase? Well, interphase, the cell is big. So telophase, if we find two little cells, maybe they have just divided.
Oh.
That's telophase. I think. Educated guess? All right. What is this one?
Meta.
Looks like meta. There's early anaphase. Look at that. All right. Okay. And if you take onion roots, if you grow an onion, you know when you put them in a glass and they grow roots,
Yes.
and you take the little root and you squash it and put it under a microscope slide, and can you see this? Can someone please remind me? Or someone, yeah, take an onion, let it grow roots. Okay, we'll try and
Just
see
in a
it.
glass with
I've
water.
actually got prepared slides in the middle room. You must pull that out. Can have a look. Okay, so yeah, there they're all together. Animal cell, look how beautiful these are.
Yeah,
They stain up so pretty. So what stage is this?
Interphase.
Yeah, I'm not sure. It's interphase going to prophase. Okay, maybe that's interphase, or what's this?
Prophase.
Maybe, yeah. So it's hard to, or maybe there, hey, look.
Yeah.
That's prophase. So that's interphase. It depends how they take up the stain. There's prophase. What's that?
Metaphase.
What's this?
Anaphase.
What's that?
Anaphase.
Later anaphase. What's that?
Anaphase.
And then maybe little ones. There, look.
Yeah,
Telophase and cytokinesis, because now they're smaller. All right, and these are just other pictures. So prophase, maybe early prophase, late prophase. This is?
Metaphase.
Okay, they're
Anaphase.
nice and ordered.
Anaphase.
And then?
Telophase.
And?
Cytokinesis.
And there, that's what it looks like. These little, I think I showed you this video
Yes,
where they
yeah.
were looking like worms, and I said, it doesn't look like the way we draw it. I mean, those are the chromosomes, and these are the organelles and the mitochondria, and, okay. And then that's a different kind of photograph outside the cell. So, I mean, look at the cell membrane. It's all these
That's
things on it,
crazy.
and it's dividing, yeah.
We always draw it like perfectly, but it's not.
But
Yeah.
we always do that, hey?
Yeah.
And there's that cell now, it's dividing. The zygote is dividing and it becomes an embryo. There's the little embryo and it's surrounded by a protective layer.
I always thought embryo was one cell. That makes
Okay,
sense.
and I'm not going to do this now. Right. Are we happy now?
Mitosis almost done.
Oh,
There's little bunnies on top of me.
All right, so what I was going to do is, should we go over the mitosis 2 worksheet?
Yes.
Because I've got it uploaded here now, because I've already gone over my mitosis 1. If you find problems there, I think then come and ask me tomorrow. Okay, so let's go into Google Classroom. Here. Okay, so Priyasha, it's in Google Classroom. The Mitosis 2 worksheet is uploaded into today's lesson. So it says Wednesday the 24th. Yeah. Okay, I'll try and make it as big as I can. Oh, but this is the model. I want the model. It's got that in there. I uploaded both of them.
Jane, don't vote for the cane.
Okay. Oh, all right, so here's the model. Here, this thing. Okay, so here we've got 6 cells and they're in various phases of the cell cycle. Note they're not arranged in the order in which mitosis occurs, and one of the phases of mitosis is shown twice, because we said early, late, whatever, and then you're going to answer the questions. Where's mine? I wonder if it's at home, because I kind of need to see the questions too. Okay, I'll just move it up and down. All right, so explain well, we've done this, okay? Explain 2 reasons why, do it this way. Explain 2 reasons why mitosis is important. So from this morning, mitosis allows for growth, or like the strawberry plant. The strawberry plant?
Grow asexually.
Asexually, vegetative reproduction, make little clones. Just like at the back there, my spider plants. Okay, that's why I keep it here. And those desert roses and geranium. All of these can grow vegetatively. Yeah, okay, this is green one. Even this, and the spider plant. Okay? All of these can grow vegetatively. You don't need seed. Okay, so this is called geranium. And if I'm lucky and I break a branch off and I put it into water with a bit of rooting powder or something, it will grow into a new plant. This is called spider plant. There are other names. Alright, and that little baby there, if it breaks off, it's going to grow its own roots and grow into a new plant. But they're clones. Vegetative asexual reproduction. This one is in the tank at the back there. I water it twice a year, if I remember. It's a little water cycle. It just grew and went mad and I left it. So if I plant this in the ground now, it's going to grow vegetatively, make roots and things. All right. Okay. So for growth and to grow vegetatively, and what else did you have?
Repairs,
For?
like reparation of tissues.
Yes, repair of tissues, like if you cut yourself. What else? What else?
I said exactly the same as you.
Okay, and what about little bacteria dividing or amoeba?
Aware of unicellular organisms
Yes.
to reproduce.
So growth of organisms to make cells. Because you see, I said explain 2 reasons.
Yeah.
Growth, make more cells. You can say it in other ways. Just try and make this a bit bigger. Yeah. Helps repair wounds by making more cells to replace the damaged ones, like these cuts on my hands. And then asexual reproduction, like when amoeba or bacteria divide, or when my little spider plant makes more babies, or this geranium, if I break a branch, it grows into a new plant. Alright, so now this is the one you were struggling with. How many DNA molecules are present in cell C? Well, how many chromosomes are there here?
8.
How many chromosomes?
4.
But
Oh,
they
sorry.
replicate.
DNA molecules is the
8.
There's 8. There's 8 chromatids.
Sits.
Alright, then how many are there in cell D?
I said 8, but it's 4.
Yeah, because there would have been 4 strands of DNA called chromatin. Okay. So the diploid number of the cell is 4.
So is cell D in interphase?
Yes, and you can't see the 4.
Oh.
You're looking at the ends of the DNA molecules because it's been cut.
Yeah, okay.
They're not lining up and pretty like we draw them. Okay, so E, how many?
8.
DNA molecules, yes, because there are 8. What are they called?
Chromatids.
But now because they've been separated, they're also called daughter.
chromosomes.
Good. Alright, then I can't remember what's after this. D, cell F, how many DNA molecules?
Did someone have
Okay, how many DNA molecules?
8.
How many chromosomes are?
4.
Replicated chromosome. Hi, yes.
Yesterday we went to get some. Our friend when
Yeah, we finish at 12.
Yeah.
I'm saying the
Oh,
gas is not
oh, the
clear.
gas. Okay,
Yes.
that's fine. I'm
When,
warmer
when,
now.
when they
Okay.
bring it, then I'll put one for.
I'm warmer now. This morning I was like this.
Okay, yes,
I'm
okay.
okay now.
Yes, but
Something.
I'll bring like
No problem.
when they come, bring them.
Okay, cool. Thank you.
Thank you. Bye, sir.
Okay. All right. Cells A and F show an early and a late stage of the same phase of mitosis. Okay, which phase? So A and F.
Prophase.
Yeah, so there the chromatin threads are starting to coil, there the centrioles, and I could say why, give evidence. So don't just write everything you learned off by heart, look there. The centrioles are starting to move apart. I've lost my stick. Okay, I need a laser, mine is broken. Alright, so the centrioles, oh, remember the centrioles. So at the in interphase, there's one pair of centrioles. Little microtubules, they've got a really fancy structure. So then during prophase, they replicate. So you end up with two centrioles. They replicate. All right, and then they move to opposite poles, and the spindle fibers develop between them. So that's centrioles, and the little patch of cytoplasm where they are, then we call it a centrosome.
Mia.
So,
Oh, sorry.
all these words, all these words. But then they replicate.
Sorry.
No, it's fine. So then they replicate, and they're going to form the spindle fibers between them. Okay. Where are we now? All right, so they both are prophase. So there you've got, A is earlier prophase and F is later because the chromosomes have replicated and the chromosomes have replicated and the nuclear membrane is broken down. And here the centrioles weren't even at the poles, now they are. All right, so next one. Which cell is in metaphase? C. Which cell is starting karyokinesis?
B.
Karyokinesis. Wait.
No,
I need the question. Oh, completed karyokinesis. Yes, B, because you've got two nuclei. Sorry, I'm moving up and down because I don't have mine
We're
here.
starting to.
Well, you need to see the answers anyway. Then, is starting to undergo cytokinesis.
What? No.
Is starting to undergo cytokinesis.
Oh, yes, it is. Look at the edges of the cell.
So which one is it?
Inner. E.
Oh,
Oh,
yes.
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 I think it's both.
Well, this one's starting, this one's further. So it looks like it's E. Did I have it right?
Yes, you did.
Oh, I did. Okay. In cell A, what is the structure labeled X? That. Centrioles, the two of them, or the whole thing is a centrosome. It's a centrioles.
So can we say century?
All centrosome.
Okay. So there would be two
These-
centrosomes.
Centrioles. But the whole thing is a centrosome. So just say centrioles. There's two.
Okay.
Okay. All right. Then, okay, place the diagrams in order from first to last. So let's look here. So first is, yes, Priyasha?
What a centrosome is?
Okay, so centrioles are little microtubules. They've got a really pretty structure. They don't look like my pen. So these are two centrioles. They are microtubules. Like little, okay, I don't want to go into too much detail. But together, they are called a centrosome because they're always found in the same little patch of cytoplasm near each other. Centrioles, two of them, but together, in their little patch of cytoplasm, they're called a centrosome. I don't really use the word centrosome.
Okay, thank you, ma'am.
Okay.
So then, ma'am, if they said, what are both of them, could we have said two century?
Two
So-
centrioles making up the centrosome.
Oh, okay.
Yeah. It's like I said, I don't really use the word centrosome often. All right, so order. Which is first?
D.
D, then
A.
D, A, F,
C.
C, E, Daf Kib. Okay, so if I was marking, I would remember Daf Kib. All right,
And is it okay if we just put little arrows in between?
you don't even have to look at mine. Okay.
I'm just on another level.
See, now this, when I did this worksheet, I thought, I'm leaving another one, but it actually had different types of questions. So I don't mind, you go and look at number one, We can see if we need to do anything there. So, the nucleic acid that occurs in the nucleus. DNA is a
nucleic acid.
DNA, and then the other one is RNA, and it makes the ribosome. Okay,
Yeah.
so it's DNA. The structure that connects two chromatids, we've done this ad infinitum, so that is the centromere. The structure which forms in prophase along which the chromosomes move. I know I didn't mention that in the videos.
Spindle fibers.
Yeah, so all those fibers they call the spindle.
Oh.
But I know, if you said spindle fibers, it's fine. The one half of a chromosome. Chromatid. The organelle which anchors the spindle fibers in animal cells. Or centrosome. See? And then the part of the chromosome which attaches to a spindle. Oh, it's not here.
It's the centromere.
Centromere. Maybe it's on the next page. There it is. All right. Name the phase of cell division which best fits. So the sister chromatids move apart. A for anaphase. Yeah, A for wave. Anaphase. The nucleolus begins to fade.
Prophase.
Yeah, can you see? Prepare. And I know the answer here. A new nuclear membrane forms around both sets of chromosomes, actually. Telophase. Two nuclei. The cytoplasm of the cell divides into. That's wrong.
Yeah.
That's wrong. What is this supposed to be?
Cytokinesis.
Yes. I think I read it as the chromatid sequence. So this is cytokinesis. I'll correct it. And then the chromosomes are located at the equator.
Metaphase.
Metaphase for middle. And the nuclear did I have it? Oh, nucleolus. The nuclear membrane phase, so prepare. Prophase. Cleavage. Now they talk about cleavage furrow. So when that cell starts dividing into two, all right, that region there is called the cleavage furrow. All right, like that. Okay, that little region there. But we don't need to know that. I mean, there's a whole science behind it.
So, ma'am, I said actually oh, never mind.
I didn't ask you. I said the division or cleavage when the cell starts to split into two. So did what did I say? Telophase. Why did I put that?
I also put that because it says that in the textbook.
But when the cell starts, the cleavage furrow means the cytoplasm dividing. It should just be cytokinesis.
Yeah.
Okay, I must change that. The chromosomes move towards. What was that? Did it go dark?
No, it was a notification. Sorry.
Okay, the chromosomes, because it reflects it, the chromosomes move apart, move towards, apart. Spindle is being formed, those little fibers from the centrioles, and then the chromosomes are replicated. I said you're late anaphase or early prophase. Because the cell doesn't say, okay, start this, stop that, whatever. Okay. All right. Did you get these right, mostly?
Yeah. Except the 3.71.
Yeah, I must just go with yeah. And I must change those two. The period of cell growth and development when it's not dividing into a phase, when it's making proteins. And then I said make a neat label drawing of a plant cell which has 6 chromosomes, da 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 da. Okay, so let's draw it and then we're done.
I forgot the heading.
After break?
Yeah, break off.
Double English.
And then maths.
Is it break off?
No, it's
Anaphase.
Oh, sorry, double Afrikaans.
Guys, it's break off.
How many must I draw here?
6.
6 chromosomes.
I mean 3.
But there's 6 chromosomes. So there's going to be 6 replicated chromosomes that,
Oh,
but there'll be 12
DNA molecule.
all chroma.
I did this so wrong.
Must I draw anything at the pole?
No,
You just do that if you want, like a little star, but you didn't know that.
I only did 3 for some reason.
Because you thought each DNA molecule is a chromosome, but each DNA molecule, once replication has occurred, is a chroma. Alright, so heading, diagram to show plants are undergoing anaphase of mitosis. So maybe sometimes you say draw a neat labeled plant, a drawing of showing the chromosomes being moved apart. So then you've got to say it's anaphase. Yeah, I told you it was anaphase. Okay, so you'd get a marker saying anaphase. Alright, so we would label chromatid, I'm just going to abbreviate, spindle fibers, okay, nice neatly below each other, and oh wait, why have I got centros? Mustn't have that, eh? Okay, I obviously did this quickly. So forget that, but any other labels, plant cell wall, cytoplasm, whatever.
So, oh, I said chromatid, yes.
Okay, I'll go back and correct this. And then you've got,
Cell
so when I
membrane.
mark it, my first mark, I'm going to count 1, 2, 3, 4, 6 plus 6 is 12, each as a single strand. So they're not drawn like this. Okay, they're drawn as little V's, one mark, being pulled by the spindle fibers, and there needs to be a plant cell with a cell wall. So I've made some mistakes, I see it now. Okay, are we happy?
Yes, ma'am.
All right.
Mom, can I take pictures of the drawing?
Yes.
It's all coming together, I feel like.
Yeah, you see, I knew I would have to spend time when we came back. Because mitosis is not, it's okay, but all the nitty-gritty stuff.
Yeah, like the chromatid and blah, blah, blah.
But I mean, it's still taken us much quicker than if you were in class. This is just like an overview. Okay, Priyasha, you happy?
Yes, ma'am.
Good.
Who else is on?
No one.
Wait,
Okay, I'm going to turn off the video because that was